Hi everyone, my name is Sarah and today I will be teaching you all about bioelectronic noses. Bioelectronic noses are a new way to detect odors through the air. With the idea ultimately stemming from how humans smell volatile objects, engineers found a way to measure and record chemicals. The basic fundamental is that protein receptors found on the surface of a human olfactory receptor cell is taken and are coupled to different things such as transistors and electrodes. These proteins, when they come in contact with the receptor cells, will change shape, causing some type of transformation, whether that be visual or electrical. But before I go more into detail about some of the current research and uses of bioelectric noses, it's important to understand the physiology of olfaction. Olfaction is the scientific term for smelling and is one of the oldest sensory forms of detection in the history of mammals. Also, did you know that olfactory epithelial cells are the only group of neurons able to regenerate? To break it down, all signals recorded by the body occur through a process called transduction. This is when one type of energy, whether that be mechanical, light, or chemical, is transduced or changed into an electrical signal by the body that can be recorded by the brain. Humans smell by transducing volatile chemicals floating into the air into electrical chemical signals carried down the axon and into the brain. The first step in this process is for a volatile chemical compound, or something with odor, to pass through the hairs in your nose. These are to help filter out many of these compounds as to not overwhelm the olfactory sensory system. Once this compound passes through the nasal cavity and past the nasal hair cells, it then makes it to the top back of the nose called the olfactory epithelium. The olfactory epithelium contains millions of different olfactory sensory neurons, which are surrounded by even more insulating columnar supporting cells. Here at the olfactory epithelium, the compound has to dissolve through a mucous membrane that coats this region and then bind to these receptors on the olfactory epithelium. Once bound to the human receptor cells, the potassium-linked protein channels change shape to allow for an influx of potassium and calcium. This influx of positive charge causes a potential membrane difference to reach a certain threshold, and an active potential has begun. The signal is carried on the long axon of the receptor cells through the ethmoid bone and into the olfactory bulb of the brain. However, each olfactory neuron can only really record one type of stimulus or smell. So alone, the olfactory neurons would have a hard time discerning the different types of smells found in everyday type situations. Because the human body is so cool and efficient, it figured out a way to solve this problem. Different olfactory sensory neurons converge with other olfactory sensory neurons into a structure called the glomerulus, which then also converges into a mitral cell. This mitral cell then relays the signal into the brain. Different combinations of olfactory sensory neurons and mitral cells can then be combined to be able to form all 10,000 different smells that we humans can pick up. The best analogy for this is to think of this in terms of music. Each combination of mitral cell and olfactory cells is like a musical note, and the smell of any volatile object can then trigger many of these combinations. In this sense, the olfactory epithelium is kind of like a super large piano that can help create thousands of different musical chords. When these different chords or sensory combinations are sent to the brain, they actually take two paths. One path is to the frontal cortex that processes these scents, and the other is to the limbic system where all our emotions are processed. This is to evoke the sympathetic fight or flight response to help us quickly get out of danger, like if we were to smell smoke. Now with a basic understanding of how we electrically detect chemicals, it'll be easier to understand the basics of a bioelectronic nose. Like I said before, researchers and companies take the potassium protein channel cells from different human olfactory receptors and bind these to create an electrical signal. Different researchers are choosing different human olfactory cells to test, and then use different methods to either measure or detect when a chemical compound has bound to one of these receptor cells. In these next couple of research articles, they will give more light into what is being done and to test and visualize smell. One of the hard things about making a humanistic bioelectronic nose is that these sensors have to be very small and have previously been done inefficiently by measuring the change in mass due to the absorption of the stimulant molecules. The sensors commonly used are frequency detecting sensors such as quartz oscillators or surface acoustic wave devices, which pick up the changes in frequency related to the mass change. One of the current studies was conducted on interdigital electrodes that have been covered with PC lipids. They are able to give responses to odorous substances such as acetic acid, eugenol, and methanol, but give no response to odorless substances like carbon dioxide, and liquid paraffin. They can also respond in room temperatures within 2 to 5 seconds. These interdigital electrodes are made using traditional semiconductor technology and are made with a few different layers. The base layer is a silicon-oxygen compound with an interdigital electrode in the center with a lipid membrane on top. The width of the gold strips is about 50 picometers, with the gap between these gold strips being also 50 picometers. The active electrode area is 3 millimeters by 3 millimeters. The membrane casting for these electrodes start by dissolving the lipids in a suitable organic solvent, and then the lipid solution is injected into the surface of the interdigital electrode with a microsyringe. After evaporation of the organic solvent at room temperature for 4 hours, the sensor is now ready to be used. To test these interdigital electrodes, purified air is first put into a steel balloon and is used as a carrier gas. This is then passed through a saturator which contains alcohol. The temperature is kept around 25 degrees Celsius with the dew point of the carrying air at negative 30 degrees Celsius and the relative humidity at 1%. The total flow rate used is 200 milliliters per minute, and the linear velocity is 2 centimeters per second. To begin the experiment, the purified air from the balloon flows through the gas chamber in which the sensor is suspended. When the resistance of the sensor becomes stable, the two flow meters are regulated in order to get a proper ratio. First, the carrier gas with alcohol is passed to the waste until the ratio has been established. Then the three-way valve is turned on so that the alcohol-air-gas mixture passes into the chamber and the sensor shows its response. When the resistance of the sensor becomes unchanged, purified air only is then again passed and the resistance of the sensor recovers to its initial value. It was found that all lipids possessing a trimethylene polar head can give a good response when detecting alcohols, while PE and PS lipids gave poor results. Also, there was a common behavior for the responses of the 7 types of lipids tested to the 8 alcohols, and all the curves are linear functions. As mentioned before, the response of the lipids to odorants is brought about by changes in the phase boundary potential, or the charge separation between the two interfaces. Basically, this is when odorants are absorbed on the lipid membrane, which then in turn causes a conformational change of the membrane. Then the orientation of the fixed charge of the membrane is altered, and in turn leads to a change in the phase boundary potential. Also, for dry membrane conditions, the fluidity of the lipids is decreased, so the molecules aforementioned rotate with difficulty. This is really interesting because not only can we get lots of different applications from this, it also helps us to understand how the human olfactory system works. This supports the idea of how potassium channels work in the human body and allows the human race to understand physiology even better. Another major research in the field of bioelectric noses is studying different specific human olfactory receptors and then how to measure or visualize odor detection. One study conducted looked into using a single-walled carbon nanotube field effect transistor, or an SWCNT FET. This will cause a measurable electrical signal once an odor is detected by the SWCNT FET. One of the desired outcomes of having a humanistic bioelectric nose is to be able to measure, record, and reproduce olfactory results. To create these transistors, essentially lipid membranes that contain human olfactory receptor 2AG1 or HOR2AG1 were coated onto the SWCNT FET surfaces, and then the deformation of the HOR2AG1 protein upon binding to the specific odorant molecules was detected by the SWCNT FET. An OTS self-assembled monolayer with non-polar terminal groups were patterned onto a silicon dioxide surface using photolithography. When the substrate was placed in the solution of the SWCNT, a single layer of the SWCNT was selectively absorbed onto the bare silicon dioxide region. Electrodes made out of 30 nanometer silver and 10 nanometer palladium were made using conventional photolithography as well. The HOR2-AG1 protein was expressed in E. coli as a fusion protein with a GST tag at its N terminus. Then, the cells were sonicated to obtain a membrane fraction. To remove all the other membrane-integrated proteins except for HOR2-AG1, the membrane fraction was treated with 25-triton X100 and then collected by centrifugation. 1 micrometer of the collected fraction was then evenly spread onto the SWCNT FET and then vacuum dried for 4 hours to immobilize the HOR2-AG1. Olfactory receptor molecules, as part of the GPCR family, usually bear ionizable cysteine residues and exist in conformational equilibrium between inactive and active bipolar states. So while the human nose is constantly smelling the same odorants, desensitization to these odorants occur over time and are no longer noticed by a person. Once a specific odorant binds to the receptor equilibrium shifts to an active state with a negative charge. These then cause negative electrostatic perturbations on the CNT junction because olfactory receptors in the active state produce a negative charge. This is one of the first demonstrations of monitoring the operation of G protein-coupled receptors. One of the other goals for bioelectric noses is to be able to visualize odor detection for security and protection applications. This study conducted looked into coupling an ion channel to the human olfactory receptor to directly detect odorant binding with the HOR. The KIR 6.2 channel protein is a subunit of the ATP-sensitive potassium channel, and 4 of these molecules form a potassium-positive selective pore. The ion channels were coupled to the human olfactory receptors to directly detect an odorant binding with the OR or olfactory receptor. The response was then visualized using fluorescence image scanning and using a fluorescence imaging plate reader flipper membrane potential dye. The human KIR 6.2 potassium channel was coupled to 4 different HORs and were then expressed. After 48 hours, the protein expression was confirmed by immunocytochemistry and Western blots. To measure the function of HORKs as an odorant sensing material, the flipper membrane potential dye was measured by a spectrophotometer. The fluorescent change of the membrane potential dye indicated the flux of the positively charged ions. The fluorescence intensity increased as the positively charged ions flowed into the cell and decreased as the positively charged ions flowed out of the cell. The fluorescence charge was negligible when cells without the HOR2K was stimulated, indicating that ion flux was then generated upon ligand stimulation. The HOR2K was very specific to the amyl butyrate among similar butyrate compounds, which was expected to be similar to the previously mentioned study. One advantage of using these HORKs as odorant sensing molecules is that the sensitivity can be increased by the higher influx of ions via the coupled potassium ion channels in addition to the calcium ion influx. It also allows for detection of a lower concentration of odorants. The patterns of odorant binding with HORKs can be rapidly visualized using a membrane potential dye, allowing for the visualization of smell. Bioelectronic noses are currently a hot topic to research. With several different possible applications, not only do they help us study and understand human anatomy and physiology, it has several other safety applications. For example, in the food industry, bioelectric noses can be used to smell different foods for possible contaminations. They can also be used for fast screening in airports or other areas, detecting toxic gases, or even potentially as a diagnostic tool for diseases. These are the resources I used, and thank you so much for watching.